Welcome to Nikolai's genetics lessons, and today's video is going to be very interesting because very unusual results, but anyone can get them. So here's a question which I got yesterday. Why do DNA samples migrate in the opposite direction while the ladder marker runs normally? So take a look. Here's the gel, here's the wells here on this side, and ladder moves in this direction, but you do not see any bands here. You see them all here moving in the opposite direction. And here is a description of the situation. I'm experiencing a very confusing result with my 1% Agarose gel electrophoresis. Here are the details of the run. 10 samples. Setup. The gel was run from negative to positive as standard. Observations. The DNA ladder marker migrated perfectly in the correct direction towards the anode or positive. However, all my DNA samples migrated in the opposite direction towards the cathode. So it's here. All the samples ended in the wrong direction. This is indeed interesting and unusual result. In gel electrophoresis, we rely on the fundamental physical principle that DNA possesses a highly negative net charge due to its phosphate backbone, forcing it to migrate towards the positive anode. Because DNA ladder migrated perfectly in the correct direction, we can immediately rule out any hardware issues such as swapped electrodes or global gel preparation errors such as casting the gel in a water instead of buffer, and the phenomenon of samples migrating in the opposite direction while the ladder runs normally is a sample-specific charge or physical transport issue. So the question is, can specific contaminants cause charge reversal? Yes, absolutely. This is the most chemically sound explanation for a selective directional split. For a DNA sample to run towards the negative cathode, its negative charge must be fully neutralized and inverted into net positive charge. This happens via two primary mechanisms. If, for example, your DNA extraction was incomplete, the sample may contain higher concentrations of basic proteins with higher isoelectric points, pi over 9.0, such as histones, protamines, lysosomes, or certain cellular debris. At a standard running buffer pH of about 8 to 8.3, these basic proteins carry strongest net positive charge. If they remain highly bound to your sample DNA, they may fully coat the nucleic acid backbone, and if positive charges of the bound proteins outnumber the negative charges of the DNA phosphate groups, the entire protein DNA complex gains a net positive charge and migrates towards the negative cathode. And second explanation can be presence of the cationic detergents or polymers, which cause charge inversion. If, for example, your extraction protocol involves cationic agents that were not completely washed away during the final purification steps, they will complex with your DNA sample. So what would be troubleshooting recommendations? In order to resolve this issue and salvage your samples, try the following steps in your next run. Perform a proteinase K digestion. Treat a small aliquot of your sample with proteinase K 1 to 2 micrograms at 37 degrees to 56 degrees of Celsius for 30 minutes. This should be done prior to loading, and if basic proteins are causing the charge reversal, digesting them will free the DNA and restore its normal negative charge and forward migration. Second recommendation would be to repurify the DNA, perform an ethanol precipitation, or pass the sample through a commercial silica column cleanup kit to remove residual proteins, salts, or CTAB detergents. And the last recommendation would be use fresh buffer, thoroughly wash your electrophoresis tank, and cast run the gel using freshly prepared pH verified 1 Ti or TBE buffer to eliminate electroosmotic instability or pH drift. Hopefully these advices are going to help you to get normal expected results when you run a gel next time. And this is all for today. Subscribe and see you in the next video. Goodbye.